Thanks. I'm happy to introduce Mike Miller, who's an old friend and pretty much a founding member of the field of computational neuroanatomy. I could spend the next hour telling you about Mike's accomplishments, such as his derivation of a proof to construct geodesic paths on the manifold of nonlinear warps, which resulted in his quite famous LZD-MM algorithm. But instead, I'd like to talk about something else about Mike's character, which is, I think we've all been to scientific talks where at the end of a talk, someone raises their hand and asks a question of the form, this talk is vaguely related to something I presented in 1972, and I'm very disappointed you didn't talk about that. And Mike is the opposite of that. Mike is the guy who, at the end of the talk, springs to his feet and says, I love this work. And this is personally gratifying, but I think it's also a fundamentally different model of science that favors collaboration over competition, and is one of the things I really appreciate about Mike and his contributions to science. And today, Mike is going to talk about integrating biological information across the many scales of the human brain. And this is, of course, a central interest to our P41. And so I think we're all excited to hear the talk. Okay, Mike, it's all yours.
Good. Is there any feedback, or are we okay? Is it okay?
Yeah, we hear you well. Thank you. Okay. And your screen.
Let me make sure that I'm muted on this machine. Oh, I am. Good. Well, what a wonderful introduction. And thank you for making that statement about my character. I would like to believe that's really true. Thank you, old friends, at a wonderful place. I always say Hopkins is great, but there are other great places, and this is one of them. I'm going to be talking about representations of the brain, unifying tissue and molecular scales. This is a talk that I haven't yet really given in total, so we know how those kinds of talks go. So this is biomedical data science, I believe, and I'm going to make that clear, I think. So I'm going to do some things that are very simple for members of this audience because they've invented many of these methods. But I wanted to make the point that for our community, biomedical data science is really about bringing mechanistic models and data-driven models to biomedical data. So that's what we'll see today. So I'm going to talk about these topics if I get through them. I'm going to talk about the theory of transformations and diffeomorphisms. I'm going to talk about multi-scale computational anatomy and LVDMM. I'm going to talk about the face of Alzheimer's disease. I'm going to talk about soft matter geodesics. And I'm going to try to do the pointillism of molecular and tissue scales. And we're going to do it by unifying these scales with generalized functions. And I'll try to make that clear if I can. The brain is a massive multi-scale treasure trove of data science, of data and science. I don't have to tell you all this. I got this from Brian Caffo. I like it because it sort of shows a nice, we have the brain in the middle, and then we have in FreeSurfer or Mri Cloud, we have 300 structures at 1 millimeter corresponding to a data reduction for maybe 2 million voxels for MRI, which of course is 100 billion things at 1 micron. We're all now working at the micron protein cell scale. We in particular are interested in the medial temporal lobe. So that's one of these 300 structures. It's hippocampus, amygdala, entorhinal cortex, transentorhinal cortex. So these are all the structures. And of course, there's more detail as we go to electron microscopic scale. We in particular are trying to put together histology and proteins and now spatial transcriptomics with mesoscale imaging, as well as Highfield Mri, as well as clinical MRI, because ultimately for neurodegenerative diseases, we want to take the disease, which we measure in proteins, and map it to surrogate markers, which we can measure clinically. And I'll tell you about some of that. While information in the brain is clearly billions of bits, a conjecture is that disease will likely live in a low-dimensional manifold. And I'm thinking of David Mumford and Alan Newell's paper, the phase six, the seven faces of Eve. Essentially that, the face for all of its voxels lives in about a 7-dimensional manifold. Brain mapping allows us to discover hypothesis-based discriminating dimensions, So of course, the background space is especially rich. It's millions and millions of dimensions, but oftentimes we're reduced to looking for the low dimensional representations. Now, D.R.C. Thompson was a mapping pioneer in biology, and he was extremely influential to the school that many of us come out of. And I was lucky to come out of the Grenander Deformable Template School, and Ulf knew D.R.C. Thompson's work well. And so D.R.C. used maps, of course, to do data reduction and to classify and cluster. Over the past decades, we've developed, all of us, a mathematical model of human shape and form. We call it computational anatomy. It in part, D.R.C. did many things. It in part formalized some of D.R.C. Thompson's theory of transformations. I'm gonna talk more about the theory of transformations. I think it's interesting, and I think it's good to abstract a little to see what we're doing. At the heart of computational anatomy is a positioning system. The positioning system is built on diffeomorphisms. We can think of it like a GPS. So the diffeomorphisms give you one-to-one onto maps so that we can take every point on the left densely and move it to every point on the right. And so of course we can bind information. So if we put a label on a point on the left, we can carry it over to the point on the right. And these maps have an inverse, so they're unique if you go backwards. So that means we can go over here and ask the question, what unique point did this come from? Of course, this is very important for image analysis and image understanding. Binding is extremely important. So these are one-to-one onto dense correspondences. Now we can do data science because we can do data reduction with these kinds of mapping. So here's an example. So here's an example from MRI Cloud. We now have many important softwares in the nation. One of them is a software that comes from MGH, of course. This is MRI Cloud showing Susumu Mori's atlas, which also has white matter structures. And if we can build a one-to-one correspondence to target images, which we denote as phi. Now we're gonna see that I'm gonna build phi using flows, so that's why we called them phi for flow, even though phi in Greek starts with a P. Actually, it doesn't, but flow starts with an F. Now, if we can build these correspondences, then of course we can inherit the labels on the left with all the voxels on the right. Now there are about 10 million voxels on the right here, or 10 million dimensions for these 5 or 6 modalities. So we will have done a huge data reduction. And so this is, of course, data science, looking for the dimensions that are discriminating. So I wanted to have a very simple example. So here's a simple example. So we take a brain, we can put it into essentially a service. We call this, many of us in the community call this science as a service. And then out comes a feature vector, and we can have a feature vector for every modality. So we can have, if there were 4 modalities, we could have 4 feature vectors, and in labels in the row would be all the different structures. So if there are 300 structures, there would be 300 labels. Of course, we aggregate them, so there are more than 300 labels. If there were 4 modalities, 4 times 300 would give us about 1,000 dimensions. So we would have gone from millions of dimensions on the left to thousands of dimensions here. So that's the dimension reduction. And so here's one very simple example in Huntington's disease. If we were to enumerate all the structures, so these are structures that if you know the brain, you'll know. Here's the Sylvian parietal sulcus, for example, and these are all different brain structures, 300 of them at 1 millimeter scale. Then we can look for the large deviation in the structures as signaled by red here, that indicate this is a large deviation relative to some population. And this could be a way that we could then generate hypotheses, for example. So in this particular example for Huntington's, caudate, putamen, and globus come up. I'm going to now talk about Alzheimer's disease, but I'm using this as a simple example to talk about data reduction. Now, in the 90s, our community developed a theory for computing diffeomorphisms. For those that are interested and not familiar, we are fortunate and we have a nice website now, and we can go there and we can look at many of the codes and many of the developments by our community, which is a great thing. So we can use diffeomorphisms as a prediction tool. Notice that they're changing in time. I'm going to come back to this. Time is going to be important because we essentially generate these with flows, so we can use it to predict where some shape is going. We can use it as a comparison tool, so we could take two faces, in this case simple faces, and we could overlap them. And of course, if we can compare them, we can build measurements. So here we are using it as a measurement tool. So we're measuring, I teach a computational medicine class to our juniors and seniors. And of course, they have a lot of fun making my face look even worse using computational anatomy. I'm sure if you had this at MIT, Harvard area, you'd get other things as well. But here's an example of measurement that we can think of it as the grid, how the grid deforms, which we can turn into a vector field, and we can measure the lengths of these vector fields. Now, why that's important is because we can actually turn this into a metric space. So we can build a metric space in which all faces live, and then build these transformations, and then measure the sizes of these vectors, add them up, and turn that into a metric. And I will return to this in a moment in the theory of transformations. Okay, so we're at the theory of transformations. So I wanted to read this quote from D.R.C. Thompson because it's so beautiful to think about what he was thinking about in 1917. Let me take a little water first. In a very large part of morphology, our essential task lies in the comparison of related forms rather than in the definition of each. This comparison, recognizing in one form a permutation or deformation of another, is the method of coordinates on which is based the theory of transformations, which I learned from Henry Poincaré. Now, I really like this quote. Recently, an old friend, Darrell Holmes, pointed it out to me again. I've had this quote in a paper, but what I really like about it also is that in 2000, David Mumford organized a year in imaging. It was maybe 2000, 2001, and he did it at the Poincaré Institute. I only realized that in the last year. I was at that. That's when we were doing this kind of work, and David was interested in it. And that's when I met Alain Trouvé and Laurent Younis, and we started working together over the last 30 years. I think it's 25 years. And it was at the Poincaré Institute. So I think that's a really nice connection. So let me tell you what the theory of transformations is in the setting of diffeomorphisms, because I think it's so nice. So here are two examples of tau tangle histology in the medial temporal lobe, and we'll be coming back to that in a minute. I just wanted to expand it. We pulled out the fusiform gyrus, we filled the brain with particles that we detected. And then the idea of the theory of transformations is that the object on the right is in the orbit of the object on the left under phi. So there exists some phi that will carry every point on the right to every point on the left. We can write that mathematically, which we do. And I just wanted to take a moment to talk about this. So we say that the images on the left, here's the model. I guess Ulf Grunander would have called this the deformable template model, that the images on the left can be generated by, if we call the template on the right the template, under some change, under some diffeomorphism. And we write it abstractly using dot. Now, I like dot because we can relate this to linear algebra. So if itemp were a vector, let's say a vector in Rn, and phi were a matrix, an n by n matrix, then this would just be a linear algebraic operation of matrix acting on vector. And that's linear algebra. It turns out that for human shape and form, we need nonlinear algebra, but that's we've generalized that. The whole community has generalized that. So dot just means the action. It's called the action of the transformation on the exemplar. And in the case of linear algebra, that's matrix operating on vector. I'll return to that in a minute. So that's the model. And then essentially the way we turn that into a mathematical structure is we essentially look for images under diffeomorphisms that map the template onto it, and we minimize over this large group, which we write minimize over phi. Now, I believe that Laurent and I were the first ones to prove that you can parlay this into a metric structure, a metric space on the space of all images in this model. And of course, that's been extended now to many, many different things. And I think Bruce may have been alluding to that in his comments. So thank you, Bruce, for mentioning that. Now, how do we actually do it? And how do you all do it? Because you've been doing it for years. You did it even independent of a theory like this. You did it well. We basically fill the brain with particles. And then we ask each particle to move so that when it moves, it generates a flow of coordinates. That flow moves the exemplar and then makes it look like the target. So that's how the codes work. Let's just state now the dynamical systems model of computational anatomy. It's very straightforward, and I think it's powerful. I mean, we have it, our community has it now. So the dynamical systems model of computational anatomy is that we have some images, and then we have a flow, and the flow acts over time. So that's the dynamical system. And you can think of this as the observable. This is the surface variable. And then the underlying latent variables are, we have a dynamical equation. And the dynamical equation looks like the flow is given by the Euler, what's called the Eulerian vector field, because Euler was thinking about this in 1755. The Eulerian vector field re-indexed by the flow. And so we show a picture of that. If a particle starts here at time 0, then it flows. And at every point along the path of the flow, there's a velocity that's called the Eulerian velocity, because Euler thought of this. And we fill the brain with particles. So this is true at every point, not just for one point. And that becomes our dynamical equation. So this is our dynamical system. Now, the basic idea then is flow carries image. Here's an example. Now, we're just doing this as a picture. Daniel Tward created this video. It's very visual, and it's showing us how the underlying flow, which is the latent object, is then moving the contrast, which is the observable. Here's a less trivial example. This is an actual subject from Marilyn Albert's BioCard study. Many of you know Marilyn Albert. She did a lot of great work with your group in the late 90s, and then moved to Hopkins. And we've been having a lot of fun together for the last 15 years. This is an actual individual in the BioCard study that unfortunately was suffering from dementia of the Alzheimer's type. And we're actually doing prediction here. So we're shooting the trajectory of this 3D CAD model under the dynamics that we basically estimated from real data, where flow carries image. Here, image is this CAD model. Now, I wanted to give some context because Bruce made his statement that Mike Miller doesn't like to just, talk about his thing only. I just wanted to say that theories of transformation were abounding in the 20th century. And I think it's really exciting that we were able to link to it, all of us, moving the viewpoint from the observable object, what Chomsky would have called the surface structure, to the latent underlying transformation generating it. And that is a very powerful idea, and many great people were exploring it. I just mentioned Chomsky, but I wanted to just show you this. Literally, his grammars are called transformational grammars. So transformational grammar is a theory of grammar that accounts for the constructions of a language by linguistic transformations. And of course, that was, the whole thing we learned in school of how to parse sentences. And, transformational grammars dominated the field of linguistics for a long time. Another great transformationalist who didn't need any of this mathematical theory was, of course, Picasso. And I'm going to finish with a Picasso because, he understood geodesics and principle of least action. But here's just a beautiful example of transformation. And, it's unbelievable to think about what was going on in Picasso's brain that he could create this kind of a transformation and see how this is in the orbit under possible transformations of a template. So now I'm going to turn to multi-scale computational anatomy. After COVID hit, we were all looking for things to do. I started working on this. Alain Trouvé and I and Daniel was working with us. We worked for, I'd say, 3 years on this, and we had our first written publication in CVPR where we didn't yet try to unify molecular and tissue scales. We just tried to understand multi-scale LDDMM. So I'm going to present that first, and then we're going to return to tissue scales later. So this is accessible, but we also now have a bioarchive paper, which will be coming up through the talk, because it actually really does look at molecular scales and show how to do this. So why are we looking at multi-scales? Because it allows us to really see Alzheimer's disease, not just its surrogates, and we are interested in that. So here's the cartoon scale shown here. Cartoons always look like this. Atlases are cartoons. David Mumford has a beautiful cartoon model, which is important if we want to model diffeomorphisms on cartoons. Then this is the clinical scale, 1 millimeter scale, clinical imaging. This is, of course, moving towards the mesoscale. It's really 100 micron, 200 micron, Highfield Mri. At MGH, you produce beautiful, beautiful high-field imagery. And of course, this is the micron histological scale. Now, I wanted to just advertise a little bit Caitlin Stouffer's new work with Daniel and myself, Juan Troncoso, and Marilyn Albert. This is a medial temporal lobe, a Highfield medial temporal lobe, which we have reconstructed now several. These are histology sections showing the planes. We've mapped the medial temporal lobe. Caitlin's shown here in the lower right. We've mapped the medial temporal lobe into Mypaxino coordinates. So this is really the Mypaxino coordinate system. So we can go to a page like page 35 and we can get out a section. This is the segmentation, these colors here. And then these are all the histology sections. And we're trying to put the histology back together again. And I think there's a poem about that, once you come apart, it's hard to put it back together again. But she's been putting the histology together with the MRI, and for that we've developed these multi-scale methods, which I'm now going to talk about. So let me turn to the hierarchical dynamical system. On the right is a cartoon. You will see this cartoon throughout the presentation. I'm using it to represent scales. The fine scale is at the bottom, and you can see the particulate nature. The coarse scale is at the top. The basic kinds of equations that we're writing are an observer equation where we have images at every scale, and a dynamics equation where we have a dynamical system at every scale. And we want to really couple them. And I want to just say a little bit more about why we want to couple these scales. If we think about the cortical surface, which you guys really pioneered at MGH, and think about the foliated layers of the cortex, that's really a global construct. The smoothness of the surface as a manifold is a global construct. It's not associated to the cells. So whatever we do to the cells, we want to understand that it's sitting in a larger global structure. So these scales have to be connected. And so we're trying to work on that. So here's the basic idea. We have at every scale a flow which is controlled by a velocity field. We call it the Eulerian velocity. And these velocities are coupled. So in the 90s, Don Gehman used to call this successive refinement, where we'd start with a low dimensional basis, we'd then add more dimensions, and we'd keep adding dimensions. At the finest scale, we have not only the coarse scale flow, but we also have the finer scale flow, which would have to do with the particles in their local neighborhoods. So this is the basic idea. Now, these flows are coupled, and so the total behavior turns out to be fairly nonlinear. It tightly couples the layers. So what that means is that we have at every layer an equation like this, and we can write down the kinetic energy of this system or the Lagrangian. So that would be on the right here, the kinetic energy. And so the kinetic energy would look like the kinetic energy that we used for LDDMM. That is, at every level, we have a vector field, V, and it has a norm, and the norm gives it smoothness. So the norm tells us to take derivatives on V, and the derivatives and the scale is going to be determined by the scale of the space. And then to generate any level, we have to add up across levels the vector fields. So that's the dynamics. And so it turns out that we can write down the momentum for the system. We can calculate the Euler-Lagrange equation. And then the Euler-Lagrange equation, of course, gives us the same kind of momentum conservation, and therefore geodesics that Bruce was referring to, that we have for LDDMM, but in the multi-scale setting. So it's the whole system moving together. And we can find those derivations in this paper. I want to continue showing you some ideas of why this is useful, so we won't focus on those equations too much. Here's an example. I think I may have shown this to you before, Bruce. You sent me a nice email that really made me feel good for the day, so thank you. So on the left is the single scale. On the right are two different scales from this bioarchive paper. So this is essentially the 100 micron scale, and the bottom is the micron scale. We fill it with particles, and then we take this left column as the template. We take the right column as the target, and the middle column is what's essentially mapping. Now the motions here are on the order of 1,000 microns. You can sort of see the fold moving. And so we have two flows. We have the flow of the particles moving, and we have the flow of the global scale. Now in the global scale, you can see essentially the inner and outer layers really nicely, and they're sort of moving coherently. So that's the idea of multi-scale LDDMM, to couple the motion so that things move consistently. Of course, just to relate it to things, many people have done things like this. I think this formulation is a little different though, but when we use rigid motions or when we use low dimensional motions, we often apply them in such a way that then the high resolution motions are consistent with how we apply the coarse scale rigid motions, for example. So this is extending that to the setting of high dimension. So I'm going to turn now to the neuroimaging face of Alzheimer's disease. So I want to just make the point that, we've been fortunate, many of us on this call have been fortunate that we can follow our passion, which is algorithms, computation. In my case, I like doing some mathematics, but we can really try to make a contribution to problems that mean something to people. So I've been fortunate and I've been able to do that. And of course, at your place, you guys are doing that and gals are doing that all the time. So in 2019, almost 6 million Americans suffered from Alzheimer's disease, 44 million worldwide. In, I think, 2007, 2008, Marilyn Albert came from Harvard, MGH, to our place at Hopkins, and we've all been working together on Alzheimer's disease since then. Now, in 1991, Brack demonstrated via histologic staging early Alzheimer's disease begins in the medial temporal lobe with progression of pathological tau. The Biomarker team, which Marilyn leads, and I lead the imaging core, I and others. The Biomarker team set out to demonstrate that clinical MRI could be used as a surrogate early stage biomarker. So we wanted to be able to duplicate what Brack had done in pathology, but do it in clinical MRI. So that's what we've been doing for the last, I would say, decade now. I'm going to assume you don't know the medial temporal lobe intimately. Not everyone does, many of you do. So I marked the temporal lobe and then show you a cartoon of the hippocampus, which is part of the medial temporal lobe. And now I'll zoom in. There's a layer of cells below, which is extremely interesting and important. That's going to form the entorhinal cortex and the transentorhinal cortex. And I'm going to give you a section now through this so that you can see the areas that we've been focusing on. So here's a section through the Highfield Mri that corresponds to the plane on the right. And what's nice about it is that you can see the transentorhinal cortex here, and you can see the entorhinal cortex. It virtually terminates in the subiculum and the CA1, and then ultimately think of this, all these cells as ending in the dentate gyrus, almost like a cap, like a piece of molding that fits over the end, maybe the start of the cortical surface. That's pretty exciting to think about, that we're working on that. Now, what I'm going to show you now is where Brack showed in pathology was one of the early places where Alzheimer's disease started. But first I wanted to show you this. My wife pointed out to me from Greek mythology that this is how you actually spell hippocampus. And she was very careful about the diacritical marks. So I think that's correct. And of course, seahorse monster is the interpretation here, or my translation. But you can sort of see why people called it hippocampus. You can sort of see the seahorse on the left when we annotate it. Now, this is a lovely little picture of Brack. He's pointing in his book to where in the medial temporal lobe the earliest staging shows, and that's right here, sort of the lateralmost part. Now, this is lateral entorhinal cortex. We're going medial because this is midbrain over here. Midbrain is here. And of course, it goes on up to subiculum and CA1 and 2 and 3 and dentate. Now, the fire starts here in terms of where we can visualize it in the medial temporal lobe. And so we set out to do that. Now, I wanted to show you, we specifically looked for a section in our Alzheimer's subjects, not a control, the one on the left is a control, where we could really see a deconstruction in the Highfield MRI of the entorhinal cortex and the subiculum. And notice how the contrast is like really collapsed here. It's just terrible. This is a beautiful person. And, you know, we have the privilege of working on their brain, but it's sad. You can see total loss of contrast. So that's what we measure. That's the surrogate that we're trying to get at when we look clinically. Of course, this is Highfield MRI. We don't have this resolution. That's presumably the result of pathological tau, which is causing cell death, which presumably results in this contrast change. Now, we want to map these in clinical populations, so we reconstruct this atlas. We map it onto MRI pictures like shown on the left. Each row is an individual with maybe 5 clinical scans. Not everybody has 5, some have more, some have fewer. In the Biomarker study, it's been going on since 1995. Hopkins won it, Marilyn won it in, I think, 2008 or 2009 when she went for it. And so we are still imaging subjects, and we can now look at other molecular markers as well. So what we do is we map the cartoon atlas on the right into the individuals on the left, and then reconstruct the entorhinal cortex in populations. Now, I want to just mention one result from Sue Coulison's work. I think Sue has done really nice work. Thank you, Sue. She's graduated. We were able to reconstruct the entorhinal cortex as a cortical structure, following on the wonderful work over the years that the MGH group has done on cortical reconstruction. And we essentially looked at cortical thickness within these subjects. Now we have multiple groups, but we have controls, and I'm going to show you the dementia of the Alzheimer's type group because the pictures are so striking. But these two papers were really about MCI and preclinical Alzheimer's disease. We're very interested in being able to use this as a marker years before clinical time. So preclinical means scan is years before clinical time. So here in the control group, blue means virtually no atrophy. This is the entorhinal cortex that corresponds to the population of controls corresponding to the structure on the right. And then here's the dementia of the Alzheimer's type group, and red means about 7% atrophy rate per year in cortical thickness. So that's quite a bit because this is only a two-dimensional, thickness is a length marker, it's not a volume marker. So we see about 7%. We see what's interesting about this picture, and I think Caitlin is, has more results on this now that we've reconstructed tau, that not only do we see, lateral extent, but we also see something very anterior, and that's an interesting story. And so you can see these results in 2019 and 2020 paper, where we basically talked about how we reconstructed atrophy rate per year. Now, what we wanted to do was to understand when these changes were happening. So we introduced change point modeling to answer the question of when the disease first appeared relative to clinical time. And so the basic idea of change point is depicted here. I took an example that was extreme, so it would be easy to explain change point. So we calculated the volume of the entorhinal cortex, that's what's shown on the left, for an individual. And what we see here is a large change as a function of years. Again, this is selected because it's such a clear picture. And then what we would like to do is to be able to essentially estimate the derivative. So of course, estimating the derivative introduces variance. So, it never looks as clean. So we published this in a recent paper. What's interesting about the paper is we did not only the medial temporal lobe left and right, but we also looked at multiple other markers as well as tau, amyloid, logical memory, Boston naming, multiple things. Now, but what did we show in terms of the diffeomorphometry marker of thickness? We found that if you go to clinical time and you then ask how far before clinical time is the population changing, we see in the medial temporal lobe roughly a decade before clinical time, we can statistically reliably make a statement about the population. This is not in an individual, but about the population, about a decade before the entorhinal cortex. The lateral part of the entorhinal cortex is changing statistically significantly in terms of the change point. Now, what's interesting, and perhaps a little surprising, but now we take it for granted, certainly in the Biocard group. We're seeing that tau is collecting and changing years and years before. And we're also seeing with particular sensitive markers, cognitive testing, we're seeing changes years before the shape marker in our hands currently in terms of cognitive testing. So about 15 years in this study. So we'll see where that ends up. But the important idea, I think, is that Alzheimer's disease starts well before clinical time, and perhaps this sheds light on, why the amyloid hypothesis has failed, and many of our therapies have failed. Perhaps we're just too late in the process. That will be figured out by the community. So I want to turn now to soft tissue and soft tissue geodesics. So I'm going to take us back to understanding the brain and then connecting us to molecular representation. So I want to go back to the theory of transformations and make the following point. Transformations not only bind information, giving us labels, but they actually represent the structure of the brain itself. We're representing the brain as soft tissue. So when we build these particles here and we flow them, if you look inside of our equations to build metrics and geodesics, it's telling us about what we're claiming about the structure of the brain. The geodesics are telling us about the structure. So I'm going to try to show you that a little bit. So let's define soft tissue now. So in this face, I put a little perpendicular coordinate system, and then I moved it under phi. And we notice that this axis doesn't change its length, but this axis is shortened. So we ought to be able to see that in the Jacobian. So if we look at the Jacobian of this map, that means partial phi, the x component, partial x, partial phi x component, partial y, partial phi y component, partial x, partial phi y component, partial y. This is a 2 by 2 matrix because we're in R2. It would be 3 by 3 otherwise. Then basically we have 2 rotations and 2 scales. And these 2 scales tell us about the lengths of these axes. Now, these 2 scales are going to tell us about soft. They're going to define soft. And of course, if we go all the way back to Euler, incompressible fluids, he understood this, and incompressible fluids have to do with these diagonal elements being essentially 1, not being soft. But we're going to see that in a moment. Now, soft has to do with the fact that if you take the derivative of the determinant of the Jacobian, it's equal to the derivative of this product. So if this product is changing in time, then that corresponds to soft. If the scales are both 1, then this is 0, and it's not changing its volume measure, and that's what incompressible means. So we can now ask, what is our biomarker? Well, essentially, we've built the latent variables using Euler. The latent variables are the V's. We often call them the controls. They control how the anatomy changes. And then the derivative of the log of the Jacobian is just, so the derivative of those scales, of the log of the scales, which is the sum of the scales, is just the divergence of V. So the divergence of V is going to tell us whether the system is soft or not. It's going to tell us whether it's increasing or decreasing and how it's changing. So in the incompressible case, which is what Euler did way back when, this is 0. For soft, for growth, or for atrophy, it's not 0. So that's soft. So what we're looking at here, of course, is that marker. Now, when we get into this business, you can imagine there's many, many, many paths. So we've turned a static image into a flow at which each point in time we have another image. So we have billions of extra dimensions. So this is our link to physics. This is our link to the principle of least action. And we reduce the infinity of dimensions to generate the we use the least action to reduce the infinity of dimensions to generate our soft tissue geodesics. And I just want to show you what some soft tissue geodesics look like. So here's an example of two particles that correspond to our soft tissue geodesics. And the video stopped running because it's probably running out of memory. But, oh, I can do this. Nope. Here I can show you. So this is an example of soft tissue geodesic. And notice that if these were particles in free space, Newtonian free space, they would hit each other and then pass through each other. But that's not consistent with the topology that we've put in, and that's the data science that we've put in the topology that has to do with how the coordinates, and so these two particles essentially waltz, they come together and they realize that if they're going to keep moving, they have to go off together. They have to really like each other. So these are soft tissue geodesics that correspond to that divergence that we were just looking at. And what does that mean for us? Well, on the left is essentially Newtonian particles. So they're following geodesics of free space, so they don't know about other particles. That's the left. On the right, these are all the particles know about each other through our geodesic dynamics that we've talked about. So we get really nice transformations this way. I wanted to just make the point that we're very familiar with this. We're familiar with it in different contexts. And so here is a beautiful illustration of geodesics. Of course, we learn geodesics for rigid bodies and not so rigid bodies. Here is a beautiful skating expedition where we have conservation, which gives geodesics, just like we have conservation for soft tissue. But the reason the skater is going to change velocity is because she changes her inertial operator. So let's watch her because she set a world record. I would like my equations to be that amazing, but they're not. Okay, so we are familiar with geodesics and we use them. And I wanted to make another point that they're really the ultimate data reduction because they remove all the extra dimensions. So they're really fundamental to data science. Let me finish now on my last topic. I'm going to turn to molecular scales. And I just want to say again that the artists have figured all this out. Of course, they weren't dealing with RNA and tau and amyloid, so it's a little harder for us. But they figured out how to go from micro scales to global scales. So the work I've described thus far has really been in what we would call classical physics or continuum mechanics scale. So over the past several years, we're working on how to integrate molecular scale, and that's going to take us to non-classical methods. So we talked about why we're doing this, and I think it's important not to forget why we're doing this. We really want to try to understand how surrogates that we can measure represent the true processes that are going on in the human brain, in our case, the human brain. Now, if we were to take this and explode it, what do we see? Well, here are two sections of tau and amyloid that Caitlin nicely generated for me of a particular section in this brain with Daniel Tward. And we see that we can see the particles. The particles are dense enough that they almost seem to fill and make a picture. But if we were to ask what's the data structure, because we're all computer scientists, this is the data structure that makes up these particles. And we can see that there's positions, X, Y. These are in 2D histology section. There are a bunch of features which have to do with the center, the area, the orientation, the roundedness. We can think of the features in the case of amyloidosis or tauopathy as representing the function that we're trying to understand in terms of protein misfolding. So what do we have? Well, we basically have points, and they have a weight. The weight might correspond to how many particles. They have positions. The positions are gonna relate to the resolution. And there's function. In this case, function's pretty simple. It's like orientation, roundedness, size. There's function. Now, we want to be able to do algebra. We want to be able to compute an energy like integral image squared, and then differentiate it with respect to something to take a variational problem to optimize it. We can't do that on points. And of course, Dirac understood this. So if we want to do algebra, we have to represent these points as generalized functions. So that's what we've done. So we essentially lift every point. So every point in this list, we lift to be a generalized function, which we now think of like an image. So it has a weight. We have position in the feature space of space, and we have function position in the feature space of function. Now, we have a terrible symbol here. It's not multiplication. It turns out that you cannot multiply deltas. These are delta Diracs. You cannot multiply them as we know, because we teach this in signals and systems, many of us. You can't multiply them. So the mathematicians have invented a nice symbol, which is multiplication with a circle. You can't multiply them directly, but after they act on something, after you measure them, then you can multiply them. So we should think of this as a delta Dirac measuring position of function. And so, of course, there's beautiful Paul Dirac. Look at him, just amazing. And he knew he needed these things. We need them now. And so if we want to think about space, well, that's clear what space is. We understand weights. Let's think about function now. So the functional features, we're now in a world where the functional features are many, many dimensions. It's like the electromagnetic spectrum. There's protein size, there's RNA species, there's cell type, there's tissue type, there's diffusion tensor characteristics. So these are all the functions in this representation. Now, of course, we have billions of these, so we build the brain from a sum of them. So a brain, we're going to call it mu. And I'm going to tell you in a moment why it's mu. We call it mu, and it's a sum of many of these things. Now, the important idea is you go from this table to this sum. So it's very straightforward. You can think of this as just a table represented in a form that allows us to do integration and multiplication. It allows us to do algebra. So we're ready to say what the brain as a generalized function is. So at the finest scales, we write them as mu. They're generalized functions. Think of these as particles. So this is non-classical physics. So they're like particles. Of course, at the coarse scales, we have images. The beautiful thing about generalized functions is they are also functions. So the images are classical functions, they're waves. And essentially, we entertain both of these by connecting them through this theory of generalized functions. And of course, it will reproduce all the things that we're familiar with. It just gives us a way of handling everything in a consistent manner. Now, I'm going to explain why we called it mu. And I apologize for this, that I have to explain this, but I do. Now, it turns out that we will have heard about these. Often they're called Schwartz distributions. There's many names, but there are many, many generalized function classes. You know, as soon as the mathematicians invent one thing, you realize that there are more of these counterexamples than there are of the examples. So we have functions and we learn about smooth functions, but there are many different kinds of smooth functions while there are many generalized functions. And I think the important idea that we've introduced one important idea is that we are thinking about geometric properties of the brain all the time. So we're thinking about axons, which have tangents and curvature. We're thinking about surfaces, and we're thinking about foliations, the cortical layers. We think about subcortical structures. So we really want these generalized functions to have geometric properties. So we're going to call them geometric generalized functions. But they also have another property, which is the basic property of measures. And the basic property they have is a common sense one, which is if you measure them in non-overlapping regions, they are additive. So if we take our representation and we look at it over in the cortex of one gyrus, and we look at it in the medial temporal lobe in another location, the simultaneous result is the sum of the two. That means these generalized functions are measures. Now, there are many generalized functions that are not measures. So this brings us into essentially what we call geometric measures or geometric measure theory. And so we've used that to understand how do we unify between the classical functions and the non-classical functions. So let me make it less abstract and try to show you the key modeling tool. So we have these geometric measures of the brain. That includes the classical images. So we are trying to build a representation that's consistent. So if I start with Caitlin's histology, we can then move to the Highfield MRI picture, which is a smooth picture, in a consistent manner and integrate between them. So that's the goal. Now, the principal modeling tool has two parts to it. The two parts are, so we called it mu for measure. It's a measure, just like a probability measure. It's not a probability, it doesn't add to one, but it's a measure. It has space in it, and it has the functional feature. The functional features are like RNA species, cell type, tau particle properties, amyloid particle properties. Now, there are two parts to it. There's a space density part, and there's something that we should think of like the Boltzmann distribution. There's a field of them. So for every position X, there's a conditional probability law that tells you about the functional feature, the amount of the feature as a probability measure at that X. So we call it a field of these. Now, I'm going to try to make it clear by showing you a picture that shows you space, rho, and mu X. So rho here at the fine scale, you can see rho. Notice how this looks atomic. It looks like you see particles. So rho is the density. So this is one of the modeling features in mu. So if we're going to match, if we're going to try to cluster, we can be sure that if we have mu when we cluster, it's going to cluster based on the relative density. And we all understand this. I mean, when we look at mesoscale, we often see density properties. The second part has to do with the feature. In this case, the feature has to do with particle size. So these are tau particles. And the largest ones in the histology plane are 80 square millimeters. So what color means here is at each position X, there's different mu sub X at every position. We have a probability on the Fs, and the Fs are the sizes of the particles. So, and that's the Boltzmann-like distribution at the fine scale here. Now, of course, at the coarse scale, we also have a picture, and at the coarse scale, we can actually see that if we look at the picture of the functional feature, you can sort of see the inner and outer layer as if particle size is different. We have larger particles in layer 6 than we do the other layer. So this is the principal modeling tool. And we can finish by saying, okay, let's put everything together. What is our multi-scale dynamical system? Essentially, we have functions and generalized functions at every scale. We imagine that we have a flow. It pushes the exemplar, the push, how it pushes is telling us about the structure of the space. And then we couple across levels by essentially building what we call the control, or the Eulerian velocity at each scale, by adding from lower layers, so that we could add coarser scale motions to make the dynamics more consistent when we're looking at the finer scales. And we have that at every level L. Okay, so that is our multi-scale dynamical system, and that completes my talk. I want to finish on a high note with something beautiful, and I want to show you a video of the ultimate data scientist. And it's Picasso, of course, and it's gonna go on for 1 minute and 30 seconds. You have to stay with the video because he's gonna do a phase transition at the end, and I think it's important for us to realize that how complex these spaces are. But this is an illustration of conservation, geodesics, and least action. So let's see if I can get this to run. Ah, good. Now he's setting us up for the phase transition. His brain, his geodesic is amazing. Okay, I think I'm done.
Thank you for this amazing talk. It was really nice. I just want to remind our audience that they should just raise their hands or write their questions in the Q&A box. I think we can just squeeze in a couple of questions. While they're writing, I have actually one question. So you mentioned the Alzheimer's disease that you're trying to have the biomarkers. So how much is there an individual variation between subjects, both control group and the Alzheimer's patients actually, when you compare them to the atlas? And how does it affect or how difficult it is to actually predict the Alzheimer's disease, these variations?
Well, I think it's hard enough that we don't yet have a reliable within subject test.
Okay.
So that, that tells you, right?
Yeah.
Because that's really what we want. And, so we know that we can build an unbiased enough marker that can signal something years ahead of time, but its variance, is so large that we need the population to control the variance.
Yeah, and population will be a bit difficult too, most probably.
Well, yeah, but for an individual, I mean, I don't have the opportunity. I only have me. I want you to do measurements. I want to come to MGH and have you image me, every year, and just on me tell me, what's happening.
Yeah. One more question from me, actually. Are there some other diseases that you can actually study with this method, if possible? Maybe Parkinson's disease instead of, in addition to.
Yeah, thank you. We've been also studying Huntington's ourselves, so we have quite a bit published now on that, but I think so. I think neurodegenerative diseases are natural.
Okay. Thank you very much. We are a bit over time, so I want to thank you. I want to conclude this seminar on this note. Thank you very much. Thank you very much for this talk, and see everyone next week. Thank you.